If you think chemistry is just about bubbling flasks and memorizing the periodic table, 2024 had other plans. This was the year it stepped out of the lab and into headlines, rewriting medicine, rethinking energy, and even giving insects something to worry about. In this video, we're covering a handful of breakthroughs that made chemists celebrate, regulators pay attention, and plenty of people do a double take. Before we get started, make sure to like the video so more people can see what chemistry really pulled off in 2024. Now let's begin. Some of these discoveries might just change how you see the world around you. Number 1. Lenacapavir, a new HIV defense. Chemistry often saves lives in the form of new medicines, and 2024 delivered a stunner on that front. Meet lenacapavir, a molecule developed by Gilead Sciences that's now being recognized as a promising advancement in the ongoing fight against the HIV/AIDS epidemic. Lenacapavir is an antiviral drug, but it's no ordinary pill. It's a long-acting injectable, given just twice a year under the skin. The drug works by targeting the HIV capsid, a protein shell that protects the virus's genetic material and is essential for multiple stages of the virus's replication cycle. By interfering with the capsid's function, lenacapavir prevents the virus from successfully infecting healthy cells. In 2024, 2 major clinical trials made headlines. The Purpose 1 trial, involving over 5,300 young women and adolescent girls in South Africa and Uganda, reported 0 HIV infections among participants who received lenacapavir injections every 6 months. This marked the first time any HIV prevention method had shown complete protection in that population over the course of a study. At the same time, the Purpose 2 trial followed more than 3,000 men who have sex with men and transgender women across Argentina, Brazil, Mexico, Peru, South Africa, Thailand, and the United States. It found that those who received lenacapavir saw a 96% reduction in new HIV infections compared to those given a placebo or standard preventive care. These results left scientists and public health experts practically giddy. The journal Science named lenacapavir its 2024 breakthrough of the year, citing the drug's unmatched convenience, high efficacy, and potential impact on global prevention strategies. After decades of setbacks in the search for an HIV vaccine, this is a win the world needed. For communities hardest hit by HIV, especially in regions where daily oral prevention methods are underused due to access, stigma, or logistical challenges, lenacapavir offers an alternative that fits better into real-world conditions. A twice-yearly injection has the potential to reduce missed doses and expand preventive coverage where it's most urgently needed. Number 2. RNA-based pesticides. Not all chemistry breakthroughs in 2024 were about human health. Some focused on plants and how to protect them using precision tools from biotech. One of the year's most forward-looking agricultural advances was the arrival of RNA-based pesticides. Yes, RNA, the same type of genetic molecule your cells use to make proteins, is now being used to fight off crop-destroying insects. In June 2024, the U.S. Environmental Protection Agency approved the world's first sprayable RNA pesticide, named Leadprona, developed by Greenlight Biosciences. This pesticide targets the Colorado potato beetle, a pest responsible for devastating potato crops across North America and Europe. The product contains double-stranded RNA molecules designed to silence a gene essential to the beetle's survival. Specifically, one involved in cellular respiration. When the beetle ingests the RNA through treated leaves, the molecule triggers RNA interference, effectively switching off that gene and causing the pest to die. Here's what makes it such a leap forward. Unlike conventional chemical pesticides that can blanket kill both harmful and beneficial insects, this RNA approach is highly species-specific. It only affects organisms with matching gene sequences, meaning bees, butterflies, and other non-target insects are expected to be left alone. The RNA also degrades quickly in soil and sunlight, minimizing environmental residue. According to EPA documents, Leadprona has a half-life of less than 10 hours on leaf surfaces under field conditions. The EPA's approval came with a 3-year time-limited registration, allowing the agency to monitor any unexpected effects on ecosystems or resistance development. But the green light marked a significant shift in pest control strategy. Scientific outlets and agricultural journals applauded the decision, noting that this was the first ever dsRNA product to be applied as a foliar spray, rather than embedded in genetically modified crops. Farmers in U.S. Midwestern states began pilot trials immediately after approval, particularly in areas where traditional insecticides had failed due to resistance. The Colorado potato beetle, often called the poster child of pesticide resistance, had already developed immunity to over 50 different chemical insecticides by the 2020s. RNAi provides a new mechanism, giving growers another tool in their limited arsenal. Still, not everyone celebrated without reservations. Some environmental advocacy groups expressed concerns about long-term impacts on soil microbiomes or the chance of gene silencing in unintended species, especially if RNA sequences overlap with genes in beneficial insects. While the EPA found no immediate genetic risks based on current testing, these groups called for continued transparency and post-market monitoring. Even with those watchful eyes, the approval of Leadprona showed that RNA-based agricultural chemistry is moving out of the lab and into the field. Researchers are now working on similar RNA sprays targeting leafhoppers, caterpillars, and even viral plant pathogens. For once, a pesticide that's precise, biodegradable, and molecularly targeted doesn't sound too good to be true. It just sounds like 2024. Number 3. Solid-state batteries. If you drove an electric car, or even just thought about buying one in 2024, you probably came across the news about solid-state batteries. These next-generation energy storage systems have long been seen as the holy grail of battery technology. They promise higher energy density, faster charging, improved safety, and a longer lifespan compared to the liquid electrolyte lithium-ion batteries we use today. Solid-state batteries swap out the flammable liquid electrolyte for a solid one, typically a ceramic, glass, or polymer-based material that allows lithium ions to move more efficiently between the anode and cathode. In 2024, that promise began turning into something real. Prologium Technology, a battery manufacturer based in Taiwan, officially opened the world's first GWH-scale solid-state battery production facility in Taoyuan. This wasn't another research lab or demo line. It was a fully operational plant capable of producing high volumes of lithium ceramic battery cells for commercial use. The company reported an investment of over $1.3 billion into the facility, and its initial production targets aimed at supplying the electric vehicle market by the latter half of the decade. This development was a significant turning point for both battery chemistry and industrial manufacturing. Solid-state batteries have always looked great on paper, but scaling them up for real-world deployment has been a major hurdle, especially when it comes to making solid electrolytes that are stable, affordable, and easy to integrate into mass production lines. Prologium's plant showed that, at least in one corner of the world, that hurdle was being cleared. For consumers, this could mean a future where electric vehicles charge in under 10 minutes, go farther on a single charge, and carry much lower risks of overheating or combustion. Unlike conventional lithium-ion cells, which can suffer from issues like thermal runaway and dendrite formation, solid-state batteries are more thermally stable and have a lower chance of catching fire if damaged. Throughout 2024, major automakers like Mercedes-Benz, Toyota, and Hyundai deepened their partnerships and investments in companies developing these systems, signaling serious momentum toward bringing the technology to market. Of course, nothing about battery innovation moves overnight. While Prologium's Gigafactory opened its doors, the cost of early solid-state cells remained high, and challenges, such as scaling uniform solid electrolyte layers and ensuring long-term durability, under real-world conditions, still need to be addressed. But the industry saw 2024 as a milestone, the year when solid-state batteries crossed over from R&D to real production. If this pace continues, we may look back on 2024 as the year solid-state batteries stepped out of the lab and began their path toward everyday vehicles. And when that happens, today's lithium-ion batteries might feel a bit like old tech. Still useful, but slower, bulkier, and ready to be replaced by something far more refined, thanks to some very clever chemistry. Number 4. Reprogramming enzymes. Chemists have long been fascinated by enzymes, those hard-working proteins that speed up chemical reactions in living organisms. In 2024, though, researchers pushed enzymes into entirely new territory, teaching them to do something that biochemistry itself had never figured out: breaking a silicon-carbon bond. This matters because C-bonds don't naturally occur in biological systems. Instead, they're found in synthetic materials like silicone oils, industrial lubricants, cosmetics, and medical devices, a group of compounds known as siloxanes. These materials are prized for their durability and resistance to heat and degradation, but that same stability makes them persist in the environment. Once they enter ecosystems, they tend to stay there. In 2024, a team led by scientists from Caltech's Francis Arnold Lab, in collaboration with other institutions, used a technique called directed evolution to engineer an enzyme that could cleave C bonds. Directed evolution is a process where scientists induce mutations in a protein, screen the variants for improved function, and repeat the cycle until they end up with an enzyme capable of doing something new. Think of it like putting enzymes through boot camp until they gain a new skill. In this case, the ability to tackle a bond that nature never prepared them for. The enzyme they developed was able to attack specific organosilicon compounds, demonstrating for the first time that biological systems can be modified to process synthetic molecules like silicone. While the enzyme's current performance is limited to controlled lab environments and moderate reaction rates, the fact that it functions at all is a big step. It opens the door to the idea that silicone waste, which was previously considered non-biodegradable, might someday be treated using engineered microbes. This development was widely discussed in the chemistry community because it represented a novel approach to tackling persistent environmental pollutants, not by collecting or burning them, but by breaking them down at the molecular level using biology. It's a shift in thinking. Instead of avoiding synthetic compounds that don't degrade, scientists are beginning to reprogram biology to meet those compounds where they are. The enzyme isn't ready for large-scale cleanup projects just yet, but it serves as proof that with the right techniques, even the most stubborn chemical bonds aren't beyond reach. Number 5. The AzideWhittig reaction. Not every breakthrough is planned. Sometimes chemists stumble on something unexpected while aiming for a completely different goal. That's exactly what happened with the AzideWhittig reaction, a surprising discovery that caught the attention of organic chemists in 2024. The story begins with an old classic. The Whittig reaction, developed in the 1950s, is a foundational method in organic chemistry that allows chemists to join carbon atoms through the formation of double bonds. It's been a go-to strategy for decades, widely used in the synthesis of pharmaceuticals, fragrances, and other complex molecules. At the Leibniz Institute for Catalysis in Germany, a team of researchers was working on something unrelated, developing a new phosphorus-based catalyst. But during one of the lab runs, a graduate student noticed that the reaction product didn't match any of the expected outcomes. Rather than dismissing it as a failed experiment, the group decided to look closer. What they found was unexpected. By including an azide, a nitrogen-rich molecule commonly used in synthetic chemistry, the reaction had taken a completely different path. Instead of producing the anticipated compound, it formed a new molecular structure known as a triazobutadiene. This type of scaffold hadn't been seen before in this kind of reaction, and the mechanism was unlike anything found in standard chemistry tech. Once the team understood what they had, They named it the AzideWhittig reaction and published their findings. The reaction was quickly picked up by organic chemists around the world, partly because it added a brand new transformation to a well-known class of reactions. Discovering a novel reaction pathway, especially one that builds molecular frameworks with potential utility, is a rare and valuable moment in synthetic chemistry. So, why does this matter beyond the thrill of discovery? Well, new reactions expand what chemists can build. Just like giving a chef a new cooking method unlocks new recipes, a new reaction enables the synthesis of previously difficult or inaccessible compounds. This one in particular could offer a simpler route to certain functional groups that are used in drug design, polymers, or other specialized materials. The AzideWhittig reaction wasn't planned, but it arrived with practical potential and plenty of curiosity from the scientific community. It showed that even in well-studied areas of chemistry, unexpected results can still open new doors if you're paying attention. Number 6. CRISPR gene therapy. For years, CRISPR has been a breakthrough technology in genetic research, often described as a molecular tool that allows scientists to precisely cut and edit DNA. The name stands for clustered, regularly interspaced short palindromic repeats, a natural defense mechanism found in bacteria. In labs, this system is paired with a protein called Cas9, which acts like a pair of programmable scissors, able to snip DNA at a chosen spot. That precision has made CRISPR, Cas9, one of the most powerful gene editing tools in history. In 2024, it moved beyond the lab and into medicine. After over a decade of research, the first CRISPR-based therapy was officially approved for clinical use. In December 2023, the U.S. Food and Drug Administration gave the green light to Casgevy, and in early 2024, regulatory agencies in the European Union and United Kingdom followed. The treatment targets sickle cell disease, a serious inherited blood disorder affecting millions worldwide. Casgevy works by editing a patient's own hematopoietic stem cells, which are the cells in bone marrow responsible for making red blood cells. Doctors collect these cells from the patient, then use CRISPR, Cas9, to deactivate a gene that suppresses the production of fetal hemoglobin, a form of hemoglobin that is not affected by the sickle mutation. Once the edited cells are returned to the body, they begin producing this healthy version of hemoglobin, reducing or even eliminating the painful symptoms that define the disease. By the time of its approval, data from clinical trials showed that patients treated with Casgevy had gone for months, and in some cases over a year, without serious symptoms or hospital visits. Participants who previously required frequent transfusions and lived with constant pain saw a marked transformation in their quality of life. The use of CRISPR in this way was more than a medical achievement. It marked the arrival of gene editing as a treatment for inherited disorders. Medical researchers and scientific institutions around the world acknowledged it as a major turning point. More than just treating symptoms, Casgevy corrects the problem at its genetic origin. And it's not alone. Similar therapies for beta thalassemia, hereditary blindness, and other rare conditions are already in development or under clinical review. CRISPR began as an obscure bacterial discovery in the early 2010s. In 2024, it became part of regulated human medicine. The therapy's approval reflected a larger shift in science, the merging of molecular biology, chemistry, and clinical application into a single path forward. Where older treatments managed disease from the outside, this new generation works from within, repairing the instructions themselves. Even without understanding the deeper molecular details, the impact is easy to grasp. For the first time, a genetic disorder that has long caused suffering could be corrected in a single, lasting treatment. It's not science fiction anymore. It's FDA-approved science, and it's already changing lives. Number 7. AI-driven protein folding. 2024 will also be remembered as the year computers officially joined the chemist's toolkit in spectacular fashion. The Nobel Prize in Chemistry 2024 was awarded to 3 researchers who used artificial intelligence to both understand and design proteins, those complex, folded molecules responsible for nearly all biological functions. One half of the prize went to Demis Hassabis and John Jumper, the developers of AlphaFold, an AI system created by DeepMind. Their challenge was to solve a problem that had puzzled scientists for decades, predicting a protein's 3-dimensional shape from its amino acid sequence. For over 50 years, this puzzle, known as the protein folding problem, was considered one of the most difficult in molecular biology. AlphaFold succeeded by using deep learning and training on a massive dataset of experimentally determined protein structures. In 2020, it outperformed all competitors in the 14th critical assessment of protein structure prediction. By 2021, the team had released predicted structures for nearly every known protein, more than 200 million entries, to the scientific community via the AlphaFold Protein Structure Database, built in collaboration with EMBLEBI. The impact was immediate. Structural biologists, drug developers, and biochemists began using these predictions to better understand diseases, design targeted drugs, and identify functional sites on proteins that had previously been structurally elusive. The other half of the Nobel-recognized David Baker for something equally mind-blowing: computational protein design. Instead of just decoding nature's proteins, Baker taught computers to invent brand new proteins. Think of it as designing custom molecular tools from scratch, proteins that never existed in nature, to act as medicines, vaccines, or even nanomachines. By 2024, Baker's team had already created artificial enzymes that chew up plastics and experimental proteins for future therapies. It's as if chemists got the keys to a molecular Lego set with endless possibilities. This Nobel Prize signaled a new era where AI and chemistry team up to solve problems once thought intractable. And in true Nobel fashion, it highlighted how fundamental science, in this case, understanding protein structures, can lead to enormous real-world potential. So there's our look at the chemistry breakthroughs that defined 2024. These amazing discoveries went beyond the lab and reached into daily life, improved how we approach disease, and gave scientists practical tools to solve problems that once seemed out of reach. Some are already making a difference. Others are just beginning to show what they can do. But they all started with difficult questions and pushed the science forward anyway. Which of these will have the biggest impact in the long run? And what else might be unfolding quietly right now, waiting to be noticed? Share your thoughts in the comments. We'd love to hear what stood out to you. If you enjoyed this video, give it a like to show your support, and subscribe if you'd like to stay updated on the latest breakthroughs in science. Thanks for watching, and we'll see you next time.